Hello everyone, I am Rebecca Raju of Electronics and Communication Department in SJCT Palai. So today's session will be about bioelectronic medicine. These are the contents that are included in the slides. So the development of technology is increasing to change the world. It played an important role in the development of human civilization along with cultural changes. The electronic equipment, gadgets, communication systems, transportation, healthcare facilities have provided a comfort factor in our lives. Bioelectronics is the application of the principles of electronics to biology, pharmacy, artificial limbs, the various sensors that are attached to the body, etc., and the application of bioelectronics. Bioelectronic medicine is one among them. Bioelectronic medicine uses the device technology to read and modulate the electrical activity within the body's nervous system, opening new doors to real time diagnostics and treatment options for patients. They are implanted using small devices that generates and produces digital doses of medicine in the form of electrical impulses to the nerve bundles, which creates an effect that can last longer enough to days. The dose or the range of the electrical impulses are prescribed by the physicians and doctors. So, bioelectronic medicine can also be termed as a convergent epicenter in healthcare technology and science. In healthcare, the new therapeutic field of bioelectronic medicine also draws on a wave of convergence, leveraging increasing understanding of immunology and neuroscience, experience learned in the last few decades implanting active medical devices, awareness of the side effects of pharmaceuticals, the new challenges in healthcare. It is a field that shows so much promise that it has gathered more than $1 billion in investment in the last 3 years, led by early investments from GSK and followed by other industry leaders such as Google, General Electric, and other, along with National Institute of Health and DARPA in public sector. And it is also promoted by various government agencies, academics, and pharmaceutical industry. So when we look back into the history of bioelectronic medicine, it starts in 2500 BC. People have always been intrigued by electricity and its potential for treating ailments. The origin of bioelectronic medicine goes way back to history. Researchers found that in 2500 BC, stone carvings from the Egyptian 5th dynasty gives evidence leading to the information about torpedo, which is used to uh, pain and numb. It's like an electric fish, rather like eels. This fish is capable of producing powerful electrical impulse, which will be lead uh, to the suppression of pain by placing them in the painful regions of the body. Following, it was also found that ancient Rome had also used torpedoes for the similar application. Later in 1600, Queen Elizabeth I physician also accommodated the use of electrical shocks in treatment. By 18th century, progress in electrotherapy and man-made electricity has become popular and human beings studied and discovered how to make and control electricity. By 1930s and 1940s, the neuromodulation, which is the usage of electricity to stimulate vagus nerve targeting the brain, was introduced. And in 1977, an Australian company, Lamers, developed the biphasic capacitance discharge micropulse device with equally active stimulation from both electrodes instead of just one. And in 1997, the US Food and Drug Administration (FDA) approved the implanted vagus nerve stimulation device for refractory epilepsy. By 2000, John McDonald of Washington University uses electrical muscle stimulation to exercise the muscles of a quadriplegic of 8 years. The patient defies medical science by regaining limited sensation and movement in his body. In 2002, discovery of a new biological pathway called inflammatory reflex opened the door to treating diseases systematically using electricity. By 2018, the improved mechanistic understanding of nervous system regulation and identifying lead therapeutic modalities in preclinical studies are utilized in clinical studies evaluating new bioelectronic approaches in disease treatment. As of 2022, there are there are 7,36,900 cochlear implants, 1,40,000 implants using for Parkinson's diseases, and 80,000 brain implants and 143 million of cardiac pacemakers are there in this world. The inflammatory reflects a new understanding of immunology. The inflammatory reflex is an innate physiological mechanism that the central nervous system uses to coordinate the immune response to infection, inflammation, or injury. A local protective response to microbial invasion or harm is inflammation. Because shortages or excess of the inflammatory response result in morbidity and reduced longevity, it needs to be properly controlled and regulated. Our knowledge of how the nervous system controls immune response has been qualitatively improved by the discovery that cholinergic neurons reduce acute inflammation. Similar to how it regulates heart rhythm and other important processes, the nervous system reflectively governs inflammatory response in real time. Now is the time to use this understanding to cure inflammation using reversible, selective hardwired brain circuits. Vagus nerve is one of the most important nerve in human body, which starts at the brainstem, splits into two parts and traveling through the neck, chest and abdomen. It is the longest nerve in the body and it regulates heart rate and number of other vital and essential functions. Vagus nerve is the medium for information that regulates the immune system. Then, this insight into the inflammatory response arose from studies of an experimental drug that was known to block cytokine production. It was in certain organs which was mainly administered by Dr. Kevin Tracy, a neurosurgeon at the Feinstein Institute for Medical Research in Manhasset, New York. During the experiment, he found that rather than applying the drug blocking TNF directly into the bloodstream, it was more effective on administrating through the vagus nerve. In the process, he also found that the blocking effect of TNF was lost after the removal of vagus nerve. Interested by the resultant concept, he dug up more about the content, driving him to found a paper from the researchers at the University of Colorado and University of Virginia. They had done an experiment using injected IL-1, a fever-inducing cytokine, into the body of rats but cut the vagus nerve. The rats did not get fever, pointing to the importance of the vagus in sensing inflammation. This experiment defined the scientific consciousness at that time, holding that various reflexes were carried on the vagus nerve to modulate heart rate and digestion, but not the immune system, thus leading to the conclusion that vagus nerve is the medium for the immune system to regulate its function by Dr. Kevin Tracy. Since then, numerous publications from research facilities around the world have provided more information about the anatomic, cellular, and molecular pathways involved in the inflammatory reflex, as well as supporting data for the central regulation of aspects of inflammation via these neuroimmune pathways, which have already been covered elsewhere. This numerous and diverse research have converged on two key conclusions. The first is that the activation of the inflammatory reflex, whether through nicotine or certain 7-AChR against or electrical vagus nerve stimulations, can reduce the signs and symptoms of inflammation-driven diseases in many models. The second is that altering the natural tonic control of inflammation by physically cutting or chemically denervating the vagus or splenic nerves. Now it's about biomarker. A biomarker is a measurable substance in an organism that can be indicative of some phenomenon such as disease, infection, or environmental exposure. Doctors and researchers use biomarkers to measure a normal biological process in the body, a pathological process, or the response of the body to a therapy. Since the inception of medical care, there have been interest in using biomarkers to identify diseases and enhance treatment. Indian doctors perform the procedure of uroscopy, which involves looking for disease indication in a patient's urine as early as 1500 BC. The physicians would often examine the color and sediment of their patient's urine in the 14th century and make a diagnosis based on what they saw to find diabetic symptoms. The detection of infections, autoimmune illness, immunological deficiencies, blood disorders, and other ailments is now done by doctors using white blood cell counts as the technology advances. For inflammatory reflex, there is a total of 4 biomarkers are used. The first one, a change in heart rate is the most straightforward biomarker to assess for VNS bioactivity. VNS means vagus nerve stimulation. And the variation in vagus nerve activity can impact heart rate because the vagus nerve innervates the cardiac pacemaker at the sinoatrial node. VNS can either cause the heart rate to increase or drop depending on the condition. Changes in heart rate may be a clue that certain vagus fibers carrying anti-inflammatory signals are coactivated. The vagus nerve, on the other hand, is a highly heterogeneous conduct made up of numerous diverse neuronal types with a variety of coordinated but largely autonomous functions. As a result, the diverse effects of vagus nerve activity are distinct and may differ. For instance, inflammatory reflex activation does not always result in change in heart rate and vice versa. Cytokine modulation does not always result in change in heart rate. As a result, monitoring the modulation of the inflammatory reflex generally requires more methods than heart rate measurements. The second one is by recording the cardiac RR interval and then doing power spectral analysis on the data. One can obtain a more thorough reflection of autonomic nervous system activity, which is made up of sympathetic and vagus nerve tone and their balance. These HRV analyses have demonstrated associations between RA disease activity and serum C-reactive protein levels as well as decreased vagus nerve tone. The possibility of HRV as a biomarker for closed loop stimulation of the inflammatory reflex has been considered elsewhere, although not having been verified as a method for specifically measuring the activity of the inflammatory reflex. The third one, the effects of the VNS on systemic inflammation were investigated in a number of experiments using the mouse endotoxemia model. These investigations used blood tumor necrosis factor or TNF levels or animal survival as outcome biomarkers. Endotoxin or liposaccharide, a cell wall component of gram-negative bacteria, was delivered centrally either IP or intravenously. As the spleen was removed, TNF production was reduced by 80%, indicating that splenic monocytes or macrophages are the primary source of circulating TNF in endotoxemia. Endotoxemia can be used as a biomarker for translational studies. However, it has a number of limitations. First of all, time course studies are rarely appropriate since the exogenous toxin generates a systemic reaction that can be harmful or fatal in high doses and modify an organism's baseline response to a challenge in low concentrations. Second, any tested disease model could have its course impacted by this systemic challenge. Third, it is challenging to define the use of endotoxin injection or infusion in humans and large mammals because these procedures result in systemic symptoms. The fourth one, ex bio assays in which blood is drawn and exposed to enteroxin were created by the team of Kevin Tracy and others to get around these limitations when determining if the inflammatory reflex has been triggered. The neurological set point of these circulating leukocytes, which is related to how these cells react to infection or sterile damage, can be measured by measuring monocyte responsiveness to an endotoxin challenge. The ex vivo bioassay monocyte ex vivo bioassay enables a number of successive independent assays that can track adjustments in the inflammatory phenotype of circulating leukocytes over time or in response to various treatments. The ex vivo bioassay also permits a variety of LPS doses that offers a deeper understanding of physiologic responses. For use in people, large animals including rats and mice, the whole blood ex vivo bioassay was created and modified. The absence of the central nervous system and other cells and systems that might interact with the circulating cell is a significant drawback of this bioassay. The time and specialized tools necessary to standardize the performance of a low throughput bioassay used to gauge the concentration of pertinent analytes in the supernatant are the second restriction. Now, about the simulation parameter. To design a therapeutic electrical dosage that will eventually be administered to patients with diseases, an understanding of the relationship between stimulation parameter and the immune modulating and end effects must be developed. Simulation waveform parameters such as output current and frequency are among the key determinants of functional stimulation other than duty cycle, simulation location, and electrode design, which are discussed next. So the first one will be the output current. Critical factors of the neuronal fibers that are activated and consequently the downstream effects of simulation are the output current intensity and charge density. The axonal transmembrane potential changes as charge is delivered into the nerve. Once the transmembrane potential reaches a crucial level, an action potential is produced. In earlier research, the Tracy group and other frequently used constant voltage stimulation, typically in the range of 1 to 5 volt, is used to demonstrate the TNF modulation in rodents. However, when simulating with constant voltage, the amount of current driven through the nerves is highly dependent on various electrical resistance in circuit, including the electrode material and geometry as well as electrode contact with the tissue. Although somewhat controllable in the laboratory, it is well known that impedances of implanted electrons changes over time because of tissue remodeling and electrode surface electrochemical changes. To translate the early discoveries into a consistent therapy, we first methodologically investigate a range of constant current stimulus parameters in rodents. Keeping pulse width and frequency constant at 0.2 millisecond and 10 Hz respectively, output current intensity was increased until the suppression of LPS-induced TNF production became evident above the threshold current. The empirically established threshold current ranged from 0.25 to 0.50 milliampere for mice and from 0.10 to 0.25 milliampere for rats. The various electrode geometries employed in the tests were probably a factor in the absolute difference between the two species. There was no indiscernible loss of function across a range of intensities above the threshold current, which is significant for stimulation design. Constant current of 0.75 to 1 milliampere or even 2 to 3 milliampere was used in numerous subsequent mouse trials to ensure the therapeutic threshold was exceeded. While currents as high as 5.5 milliampere have been utilized in clinical studies of vagus nerve to treat heart failure. Currents implanted on the cervical vagus nerve as of right now have designated upper limitations for output current of 3.5 milliampere. Although current was increased in controlled pivotal clinical studies in drug-resistant epilepsy to a maximum of 3.5 milliampere, the participants' maximal average output current was really only about 1.3 milliampere. The output currents beyond 2 milliampere are rarely necessary and potentially worsen patients' tolerability, according to an early evaluation of clinical experience with the device parameter. Now, in the case of frequency and pulse width, it is yet another crucial factor in determining neuronal activity, along with current intensity. The strength duration relationship is the name given to the relationship between these two variables. As the pulse width grows, the threshold current needed for activation drops. The Lapik-Orbiz equation, which was used to quantitatively model this relationship, may be solved using the two neuron or electrode-specific variables rheobase and coaxy. The coaxy is the pulse width necessary to depolarize the neuron given a current intensity twice rheobase, while the rheobase is the minimum current amplitude required to depolarize the neuron given an infinite pulse width. In order to limit the damage to tissue and electrode and prolong the battery life of a pulse generator, stimulation with a pulse width equal to the coaxin is more charge efficient than stimulation at wider pulse width. Grill and colleagues, those are the scientists who conducted a series of vagus nerve neurophysiological studies on dogs, found empirically that the coaxins of both A and B myelinated fiber were approximately 0.2 milliseconds. These results suggest that 0.2 milliseconds will be the most effective pulse width to activate the myelinated fiber within the vagus nerve. Early research on vagus nerve stimulation and endotoxemia by Tracy and colleagues reported no significant difference in the degree of serum TNF modulation between pulse width of 0.5 and 2.0 millisecond, which is consistent with our conclusion. Additionally, they discovered that there was no difference in the modulation of serum TNF between stimulation at 5 Hz and 30 Hz, although there was a bigger decrease in heart rate and the higher frequency. Subsequently, the lab of Kevin Tracy found no discernible change in the modulation of serum TNF in endotoxemic mice when activated at frequencies between 1 to 10 Hz. However, there may be situations where frequency dependency is obvious because norepinephrine released from the spleen seems to be stimulation dependent and peaks at 10 Hz, potentially enhancing cytokine regulation. It has been reported that the clinical epilepsy literature that pulse width strongly affects the tolerability of vagus nerve stimulation in clinical studies with significant improvements in tolerability as pulse duration is lowered from 0.5 to 0.25 millisecond. The typical clinical pulse frequencies for treatment of epilepsy and depression range between 20 and 30 Hz to treat RA and Crohn's disease. Subjects were stimulated with a pulse width of 0.25 millisecond and frequency of 10 Hz or a pulse width of 0.5 millisecond and frequency of 20 Hz. Next is the duty cycle. The amount of time that the simulator is on related to total time off. The two major components of these parameters being the pulse train length and the intertrain interval. These are the two main elements of this parameter. The pulse train length and the intertrain interval represent how much of the time the stimulator is on in relation to how much of the time it is off in the context of neuromodulation. In order to investigate the association between the pulse train duration and efficiency, Tracy and colleagues altered the length of the VNS pulse train during murine endotoxemia and found no difference in the degree of TNF modulation after the pulse train lasting 30 seconds or 20 minutes. Even so, the majority of the experiments carried out by their team used pulse train lasting at least 60 seconds because the inter-individual variance was higher at 30 seconds than at 60 seconds or more. This single pulse was discovered to the smallest effective pulse train in mouse endotoxemia, when this pulse train length dosing observation was later expanded, we postulated that a single supra-threshold pulse can initiate a feed-forward amplification within the spleen as demonstrated by the rising levels of ACH release in the spleen for tens of minutes after VNS in mice to explain this incredibly low trigger of the systematic anti-inflammatory impact. One unique and striking aspect of inflammatory reflex is its long-lasting anti-inflammatory effect following induction as observed in cultured cells, mice, rats, canines, and humans. Primary human macrophages in culture were treated with ACH for 1 hour, followed by endotoxin challenge for 4 hours in fresh media. Levels of LPS-induced TNF were significantly reduced from sham-treated cells at 2, 24, and 48 hours post-ACH exposure, but not at 72 hours. So in acute endotoxemia, models in rats and in vitro whole blood assay stimulation, experiments in dogs, simulation periods of 1 or 2 minutes were optimal for suppression of LPS-induced TNF production. To treat RA and Crohn's disease, subjects were stimulated with the duty cycles of 0.07% to 0.28%, that is like 1 to 4 minutes per day. So according to the disease, the duty cycle can also be varied. So now it's about the microsimulator, one of the most important part in bioelectronic medicine. So in 1965, Gordon Murray, who would later become one of the founders of the chip-making behemoth Intel, wrote a paper noting that the number of electronic components that could be added to an integrated circuit was doubling every year. This exponential increase came to be known as Murray's law. The rate of doubling was later reduced to once every 2 years in the 1970s, and with this modification, Murray's law has stood since then. To provide context, Intel's early 4004 chip had 2300 transistors, and later the Intel C5 chip launched in 2040 has more than 5 billion transistors. When lithium-ion batteries became popular in the early 1990s, they offered twice as much energy for the weight as the next best alternative over the last 10 years. Researchers have explored hundreds of new battery technologies to discover the next advance that will hold more energy, last longer, be safer, and recharge easily at a lower cost. Wireless charging using an electromagnetic field to transfer energy between two objects. The concept of this wireless energy transfer has been around for more than a century, and early work is widely credited to Nikola Tesla. Current applications of the technology charge within close proximity to an inductive charging pad or mat, but far-field wireless charging is in development in various forms without the inductive pad. These advancements, in turn, have fueled technological progresses, shrinking the size of electronics and batteries across all industries. Now, for the microsimulator of a bioelectronic medicine, especially in the case of inflammatory reflex. The use of vagus nerve to treat the indications that set point is addressing, such as chronic inflammatory diseases like RA, is not good fit for traditional cuff and implanted pulse generator. For instance, the LivaNova VNS system links to an IPG implanted in the patient's chest and uses the Huntington helix electrode on the vagus nerve in the neck. Conventional leads have delicate silicone rubber and thin wire construction that must tolerate constant flexing, particularly in the neck. To obtain the degree of relatability today expected or necessary for implantable medical devices, a viable lead may take years of design and surgical method revisions in humans. But let's have a number of limitations that have long been understood. In 1988, Loeb claimed that it would be possible to construct neurostimulators that are millimeter and submillimeter in sizing using radio frequency coils coupled to transmit power. Doing away with the requirement injected was later requirement for large batteries and related circuitry. A glass microstimulator that could be injected was later created by Loeb and Trike. It was powered and operated by an external RF coil. The RF bion was the name given to this instrument. The RF bion's two electrodes are on the opposing sides of the device and was made to be injected into muscle close to a nerve and stimulate the tissue by moving current between them. The RF bion did away with leads, but every time stimulation was required, the patient had to apply a coil to the body. Researchers at AMF, that is the Alfred E. Mann Foundation, came up with the idea of battery powered bion to address this problem. They collaborated with the Jet Propulsion Laboratory to develop a small cylindrical lithium-ion rechargeable battery that was later produced by a company called Qualion that was established especially to make this battery for medical use. The end result was a very dependable battery cell that could be totally drained all the way to 0 volt and then fully recharged. This is so-called 0 volt technology was an important advancement because it eliminates the need to remove the device from the patient's body if the patient did not recharge their device for a prolonged period. Advanced bionics developed and used the battery-powered bion technology and implanted patients for the treatment of urinary incontinence and migraine headaches. SetPoint adapted the idea of powering the implant with the third generation of the Qualion battery so that a patient does not need to worry about when to dose themselves, allowing the SetPoint implant to consistently deliver stimulation without concerns about patient complaints. Injectable microstimulator like the Bion device are not suitable for modulating the inflammatory reflex, in part because they include proximity electrodes that would not reliably trigger the inflammatory response to solve this problem. To solve this problem, SetPoint Medical came up with the idea of an integrated IPG cuff electrode. The SetPoint IPG technology was so compact that the complete IPG device could be enclosed in a silicon cuff alongside the nerve, allowing for the development of the integrated cuff. An early prototype of this integrated cuff was shown to a group of neuro and vascular surgeons at a preclinical facility early in the development of the SetPoint device. This presentation promoted SetPoint to incorporate a groove on a new microregulator device within which contacts are implanted. The entire system might then be placed inside a positioning and orientation device, a silicon support. The pod was derived from the conceptual model of a pea in a pod and positions and orients the electrodes against the nerve and insulates the electrical path to more directly and efficiently route the current through the nerve. To ensure that the microregulator remains in place, a suture is used to tie the pod closed. Now talking about the companies involved in the project, SetPoint is one of the main companies that is involved in the project Bioelectronic Medicine. The SetPoint Medical Bioelectronic Medicine platform delivers targeted electrical pulses to trigger the intrinsic inflammatory reflex. For those with rheumatoid arthritis, Crohn's disease and other chronic inflammatory disorders, this pathway holds promise as an effective alternative therapeutic option. It may also minimize risk, lower costs and increase patients' adherence to therapy. It is founded by Dr. Kevin Tracy in 2007. The innovative exclusive bioelectronic platform from Bioelectronic Medicine platform from SetPoint Medical is made up of an iPad-based prescription application, a wireless charging collar or bat, and an implantable microregulator. The miniature medical device simulates the inflammatory reflex, a natural mechanism to regulate the immune system. For a variety of autoimmune disorders, bioelectronic medicine offers an alternative therapy option with the substantial immunosuppressive side effects generally associated with conventional medicines. with the potential to grow into other autoimmune indications like inflammatory bowel disease and multiple sclerosis. Setpoint Medical has concentrated this innovative therapeutic method for creeping illnesses, starting with RA, intrinsic root. The inflammatory reflex is triggered by a little therapeutic device to balance the immune system. Setpoint Medical developed a treatment to provide a better alternative to pharmacological therapy for treating chronic inflammatory disorders that may carry less risk and cost less for patients and providers. With no regular tablets or injections required at high cost of required, Setpoint Medical's bioelectronic system may do away with the hazardous and high costs of conventional biologic therapies. To create an implantable microregulator and positioning devices, researchers have spent more than 4 years working with top surgeons. Finally, they manufactured small self-contained microregulator that produces specific electrical pulses using an integrated circuit, telemetry hardware and a rechargeable battery, which were enclosed in a ceramic and titanium cover. Physicians determine precise automatic dosing through an iPad prescription application, and patients recharge the pulse generator at their convenience using a comfortable wireless charger. The second company would be Galviani Bioelectronics. The development of bioelectronic medicine to treat chronic diseases is the focus of Galviani Bioelectronics, a cutting-edge medical research organization. Galviani Bioelectronics is a subsidiary of Verily Life Science, a division of Alphabet International Corporation, was created through a collaboration between two major global health care organizations, GlaxoSmithKline and Verily Life Sciences. GSK brings its experience in the life science and Verily expertise in software and electronics for clinical applications. Through implant-based direct regulation of particular brain signals to organs important in chronic diseases, Galvan is developing medicines to treat this disease. This treatment utilizes small effective wireless electronics and keyhole surgery to be safe, precise in their therapeutic efficiency, and minimally invasive in their administration. Patients and doctors will be able to efficiently control and optimize the therapy thanks to the connected software. Galvani has created a platform for neuromodulation with paths that are appropriate for regulating a variety of nerve targets. The implantable parts are made to be inserted using laparoscopic minimally invasive surgery in order to lessen the patient's suffering and recovering times. Galvani's lead therapy aims to treat rheumatoid arthritis by stimulating the splenic nerve. So, bioelectronic medicine is a growing field where major advancements in treatment and diagnosis are being achieved. Therapies based on neural stimulation and application of electric field are currently used to patients' quality of life. With the use of gadget technology, bioelectronic medicine may now provide patients real-time diagnosis and treatment options by reading and modulating the electrical activity within the body's nervous system. However, these therapies will still require a multidisciplinary approach to produce less invasive technique. Developments in nanotechnology and Developments in nanotechnology, biomaterials, and new methodologies will greatly contribute to this field, offering a new therapeutic tools that create great impact over the future medicine and pharmacology. This is a technology that will have a great future and profit in the market due to its advantages. So these are the references. Thank you.